Okay, so let's back up one. We did talk a whole lot about adenosine triphosphate. That's the molecule we spend for energy, and I did discuss that too, I think, in quite a bit of detail. But here's adenosine triphosphate right here. And if you back up in your notes, you'll notice that adenosine is really two things. It's a sugar, and it's also a adenine nitrogen base. Like in DNA, you've got the adenine always pairs with thymine, and cytosine always pairs with guanine, and you got double strands, and the nitrogen bases are in between, and that's what's holding the two strands together because they attract. The adenine attracts to the thymine, the cytosine attracts to the guanine, and so forth. And they can be put in all kinds of different orders, which is important. But so that's what that really is. If you want to go back and look at it, that's what you'll see. And then I have these three phosphates, and hopefully you recall that all three of those are going to be negatively charged. So because they actually all have oxygen, so they actually have 4 oxygens around the phosphorus. So that one is going to pull, because of the 4 oxygens, that one's going to pull electrons this direction. All right, that's electron. And so that will be negative. But then this one will pull the electrons even further that direction, so this will be more negative. Let's make it more negative. And this third one will pull the electrons even further that direction, so this last one is just really freaking negative. All right, so what we want to do is I want this negative particle to push away from that negative particle. Not hard. You got North Pole and magnet. Do they just like hang out together? You know, you got to put energy to make them attach. And if you let go, the magnets just go like that. So they will spread apart. So positive to positive will spread, negative to negative will spread apart. They don't hang out. They repel is the term. So what's happening here, if I can actually cause that third phosphate to push away from the other 2 phosphates, there's energy in the movement of that phosphate. And that's the weird thing to me, and I did mention that the phosphate actually moving from where it originated right here to where it will end up over here somewhere, the movement of that actually creates energy. And that's somehow the power that runs any living creature on the planet. That the phosphate pushing away from the other phosphate, that movement, the kinetic energy of that. And I said before, it's real hard for me to wrap my mind around that. It's like, really? You're kidding, right? That is the energy? Okay, well, if you say so, you know, I can buy into it. If you want me to buy into it, I can. Do I really understand it? Not really. Some of you may. That's cool. If you do, you would just meet me in my office before class some morning and say, let me tell you how that actually works. How you can have energy of motion, that moving, find energy to run a reaction or something. If you understand, it would be super. I can go down the stairs and visit with Miss Jackson and say, okay, here's the deal. So you explain to me, and she would. But, you know, we've got bigger fish to kill. You've got a lab test on Tuesday. You will have a lecture test on Wednesday, and Thursday we'll do it like we did before. There. Okay, feel better? Yeah. I'm trying to keep it away from you. Now, see, that won't be headed towards you now. That's good. You know, they're all going, it's great. That's why I was on the the discussion part. You'll do like we did before. So hopefully you're doing those online. I'm as when I have time to get to those. So a lot more grading online now that basically everything is kind of hybrid or online. It's a lot of online grading, which I'm not complaining about. I'm up for a challenge. Trust me. Give me a challenge, and I'm like, I'm there. I can handle it. I'll get it done, and I'll get it done better than most people will do it. You know, that's just me. That is me. I'm built like that. I have no idea why. I don't know. It's just genetic. I have no idea. But that's just me. So, and some people are built like that, and some people aren't. You know, and I think maybe it's just genetic. So if you're not built that way, you know, I don't know. You may not end up in a job that you just thoroughly love. How bad? You know, would that be? That would be bad. You know, I think you want to give it everything you have. I'm serious. Um, to get to whatever that is, you know, I could have gone to med school. I didn't want to go to med school. I had no interest in that. You know, I watch somebody giving blood and the blood's going up the tube, and I'm like, ugh. You know, I want to pass out. I cannot donate blood. You guys donate blood and get 10 extra credit points. And I'm like, I don't know how you do it. That is so disgusting, you know. No, I couldn't be a doctor. There's no way. But I knew what I always wanted to do. And I hope you can find your little place in the world, because you'll be really, really happy you did. But anyway, so it is a movement energy that powers the cell. And like I say, I don't understand it, but that is it. I do know that, but my head doesn't comprehend. No comprendo here. No. But, okay. So what's going to happen, really what happens is that water goes in right there. It's a dehydration, it's a hydrolysis reaction. You put water in and split that bond. It's a hydrolysis, and that phosphate will fall off. Of course, what you're left with is the adenosine diphosphate, and that phosphate, which is a PO4, is just left, and you release that kinetic energy. That's what we run on there, that little piece of energy. So it is quite amazing. It is an interesting set of events, the way that life actually works, and I think it's really fun. Very fun. Phosphorylation, that's a term, and I'll show this. If this were a PO4 group, so we'll say this is a phosphorus with some oxygen here, and some OH over here, and some OH over here, and some OH over here. This whole thing is nothing but that P, or like they've been showing, or I just call it PI, inorganic phosphate. It's all the same thing. Okay, so let's say that this marker is this thing right here, this PO4. I call it a PO4 because it tends to lose those hydrogens. They tend to pop off because they leave their electrons behind. So I don't usually say, I don't even draw it like that usually. Usually the way I will draw it will be a phosphorus double-bonded oxygen with a negative charge, another oxygen with a negative charge, to another oxygen with a negative charge. I don't put the hydrogens on there because they release without their electrons. They release the hydrogen ion. So that thing pops off, it releases the hydrogen ion. This thing pops off and releases the hydrogen ion. So you're left with a PO4 with a bunch of negative charge. That's really the way PO4 looks. So it's an acid. It tends to release hydrogen ions. It is an acid. They call it a phosphoric acid. It's a phosphate group. It's an inorganic phosphate. It's a PI. It's a P with a circle around it. It's a PO4 with the 3 hydrogens on it, or you can take the hydrogens off because they do call it a phosphoric acid also. All those things, they're all identical. And some of you are like, got it? And some of you are like, what the, did he just say? What was that? You know, if you're one of those that are going, man, got lost right there, brother, I am like, what is going on? You know, I introduced several people yesterday in lab that says, you know, hello, this is, and she has really good eggs. And if you're looking for someone to actually create a baby with, then this would be a really good choice right here. Or if you're looking for someone to father your children, this guy would be a really good choice right here. You know, I'll let you know who in class you really might consider thinking, you might consider dating, because those people are going to go somewhere in their lives. They will be successful, and they will do great things. So, do you want to be with that type of a person? Obviously, point that out. I mean, y'all are in a period where that's what you do. It's probably the most important thing that happens in your life. The whole dating event, Oh, does she like me? Oh, I don't know. Oh, and you're texting and tweeting and whatever it is going on and Facebooking and just the whole enchilada. You're doing all of it. Yeah, it's trying to establish a life. Once it's established, if you did it correctly, you're going to be like so happy. It's going to be like super. Your life will be great. If you do it badly, your life will suck. Don't let your life suck, please, please. Okay, so your father will leave the room and the instructor will show up now. So a phosphorylation is, if this were a PO4 right here, this thing, okay, and if I were to hand it over, okay, all right. Have you ever received a PO4 before? Today? Okay. Well, then she was a phosphorylation virgin, a virgin. So she is now phosphorylated. She picked up a phosphate group, So we've ended that virginity, that part of your virginity. So I knew you really had rather I didn't do that for you, but I did. I was the person who ended the virginity on that. Basically, so if you hand a phosphate from one molecule to another molecule, the one that gets it is said to be phosphorylated. It picks up a phosphate. That's all that means. So if I take, I don't know, a glucose, which is a sugar, obviously, because it ends in OSE, glucose, and I put a phosphate group on it, I add that, it becomes a glucose with a PI on it. It is now phosphorylated. It picked up a phosphate group. I didn't spell out the whole word glucose, but anyway. So it is phosphorylated at that point. That's all a phosphorylation is. And you do a lot of that. You take them off of this, and you stick them on that, and you take them off of that, and you stick them on this, and you do a whole bunch of phosphorylation events. Take them from here and move them there, and then take them from there and move them there, blah, blah. We do a whole bunch of it in biology. And you go, okay, okay, phosphorylation, go on. We get it. We get it. We're there. Phosphorylation just means pick up a phosphate group. I'm not sure anybody, and personally, that's a really good definition as far as I'm concerned. Just picking up phosphates. To gain a phosphate, PO4, you are phosphorylated. That molecule is phosphorylated. End of story. Okay, now, so for cell work, we have chemical. That would be the food that we consume. Drinks we consume. I like coffee in the morning, probably. And believe it or not, I only have 2 cups. Believe it or not, I do take 2 mood stabilizers to keep me stable. And I take 1 Ativan, which is a downer. And I take a blood pressure pill to make sure that my blood pressure is within a normal range, so my head doesn't blow apart. So I should be really chilled. I'm not. I'm not chilled. No, I had 1 cup of coffee. This is just number 2, right? I mean, I'm just, people go, oh, he's on drugs. Yeah, I'm on downers. I have to be. Imagine this without the downers. Man, wild world. I've actually been there. My friends have been through that with me. They can tell you, you don't want to go there. That's like doing crystal meth 24/7. Yeah. And it's fun. Trust me. I love it. I love it. That is so much fun to not take my medication, but I take them. Occasionally I forget. But anyway, so chemical energy, I put half and half in my coffee, at least at home. I'm not sure what that mess is we get up here, but it's half and half. So yeah, it's got sugar in it. It's got half cream and half milk. So it's got lactose in it. It has vitamin D and blah blah blah. You got all kinds of stuff in that. And I like to eat a Jimmy Dean biscuit with sausage and egg on it. I don't know, probably 4,000 calories. The one thing. So that's breakfast for me. I buy it right downstairs. Breakfast and lunch. Get a wrap so that I can take it home for lunch. Bam, bam, got them done. I got other things to take care of. Worry about lunch and breakfast and whatever. Anyway, mechanical work is just actually work that is a physical work. So we lift an automobile or a motorcycle off of a motorcycle accident victim. We can lift the motorcycle off that person and try to help that person. It's actually against the law if you don't stop and help someone. If you see something like that happen and you do not stop and get out and try to help them, you can be arrested. It's against the law. People go, but I don't know what to do. Well, do anything you can, and you will not be sued. It is your duty to do that. So don't you stop. You try to help. And there are plenty of people there, and if someone's helping in their trucks and things, and drive by. Keep on going. Don't back up traffic. Goodness. If you want to see gore, go to the web. Okay. So for transport, we need to transport stuff in and out of cells. We have active transport. We spend energy to move things into cells or out of cells against the concentration gradient. Transport. So, and in all of these, what we're using as an energy source will be the ATP. Okay. Now, the last thing they get into in this chapter is how enzymes function. What do they do? Well, we've talked a whole lot about enzymes. If they took all of the enzymes out of your body, I forgot. All of the enzymes out of your body, then you couldn't possibly function. Okay, this is row 1. I'm going to try this because I'm not liking the fact that I'm not getting to know the names of the students. So I love to know the names of my students and be able to how was your weekend? George, whatever. Just stuff, I don't know. To me, that's important. So anyway, I have this, and so this is row 1 right here. You're row 1, 2, 3. So, if you're on row 1 and you're at the end of this row, sign your name. You may have to end up signing it, well, if you're right, like your first name and last name, put the first name on top, last name on bottom. And then that way, when I look at this, then I know who is sitting where. And then, so row 2, of course, you guys, row 3 there. So we start right here. Just write your name right in the first spot. Next, you'll put yours next, and so forth. So, and then pass it on. And I'm going to try to do roll that way, and I think it helps me to get to know who you are to some degree. It's kind of hard to know exactly. And I'll do the same thing in lab also. So I think the better you can get to know the people you're working with, the better the relationship can be. And I think being successful is a whole bunch about being able to communicate. And the better we can do that, the better grades are, and so forth. So it's very beneficial. I've been in the 16th week, I always got to know my student's name, but in the 8th week, I'm just like, God, I am not liking this. So I'm trying to come up with ways where I can do that. That's for me. That's for me. It's not a negative thing at all. It's a positive, for sure. I try not to do negative things, although I do fall short. I do fall short on occasion. All right, I will admit. Okay, so enzymes, they are three-dimensional. They're third level of structure. You have primary structure, secondary structure, tertiary structure. They're all proteins. So it's a third level tertiary structure protein. It does have active sites, and the active sites actually pull molecules into that active site. Now, real quickly, I guess we can go to this screen. Need to erase that. Okay. So, and we've drawn these before, but say, for example, I typically draw mine like this. So, this is the enzyme right here. This spot right here, where the X is, that's going to be an active site. And then, of course, we have a second active site right here. We put another X, active site. Okay, so I can pull this molecule into that active site and pull this molecule into that active site in order to create a bonding between the two. So let's say this triangle molecule has a negative charge. Overall, it's got all kinds of charges, but let's just say it has a negative charge. Let's say that active site has a positive charge. Well, yes, the negative is going to be attracted into that active site. Well, let's just invert that. Let's say that molecule has a positive and this active site has a negative charge. So yes, we're going to get an attraction. Now, sometimes they call this type of an event where the molecule fits into the active site. It's not an exact fit, but it's pretty close. But when it gets in there, the enzyme changes shape so that it fits exactly. It's like trying to put a peg into a hole, you know, those children games that they do. But in this case, when you try to put it in the hole kind of changes shape so that it will fit exactly. Kind of fun. Anyway, so it's called an induced fit, to induce. It's called induced model. Enzymes are induced models. So in reality, this wouldn't really be like a triangle shape, and in reality, this wouldn't really be like a little boxy shape. But when these things get brought in, then they change shape so that they exactly fit the molecule that goes in there. To induce. If I want to induce vomiting, there really is no effort for me whatsoever. I can vomit really easily. You know, some people go, well, you're probably bulimic because you're like 6'8" and you weigh 3 pounds. Yeah. No, I'm not bulimic. But if I get too much alcohol on my stomach, you know, instead of laying in bed and have the bed go round and round and round, you know, where you have to put a foot on the floor to keep it from going round and round, you know, I don't play that game. I get up and just go, yeah. I hug that toilet, get rid of all that mess. I feel great the next day. So, to induce means to bring about vomiting. I can just do it. It's not hard for me to do. But other people, you know, they stick their finger down their throat or whatever to do that. So it means to bring about. Induce is to bring about. So that molecule brings about a change within that enzyme. So you do get an exact fit. Now, in this case, I may want to put these two molecules together, the triangle and the box. So literally, the enzyme changes shapes and it bends inward, which is kind of strange, but it does. And then we create a bond between those two. Or it would be possible instead, and I know we've been through this, so you're just repeating yourself, you old man, but go ahead, we've heard all this. All right, so what if I had a substrate which was really big, and then I had an enzyme that looked like this? You know, that whole substrate can go into this. Call it a positive negative attraction. This is positive and this active site is negative, so that positive attracts into that negative, except this time the enzyme, instead of bending in, it pushes apart. It kind of goes like that, and it breaks this molecule down so that what we're releasing are two components, that plus, say, that. You make two units out of that. You start with a big substrate and you break it into two units. Well, that's a degradation reaction. You're breaking it down. Here you're putting things together. That's a synthesis reaction. So this is synthesis, means to build. And then this is degradation. And we've covered all this in lab. But I'm real serious about, whatever I cover in lab, I don't usually hold you responsible for on the lecture exams, and whatever I do in lab or in lecture, I don't hold you responsible for on the lab test. So I try to keep those as separate as I can. That way you don't get confused. Do I need to study the lecture notes for this lab test? No, you just study the lab stuff. And for the lecture test, you just study lecture stuff. So it's a degradation reaction. There. All right. Broke it into two units is what we did. All right. So that's basically what's going on with our enzymes. That's how they work. Okay. Hopefully I cleared that. No, it didn't clear. I have to do that before I switch screens. Learn something new every day. Okay. And as we've said before, that enzyme can be reused. You reuse them over and over and over. Eventually wear out, so you have to make them again. But they can be reused a number of times. How many times? I don't know. Depends on the enzyme. Are we talking about a crayfish? Are we talking about a starfish? Are we talking about a human? Of what? What are we talking about? Where is it? How much activity does it have to do in a period of a 1 hour? How many reactions does it have to catalyze in 1 hour? 1 an hour? Okay, it won't wear out as fast if it has to catalyze 10 reactions in 1 hour. It's going to wear out more quickly. So people sometimes go, well, how often do they have to be replaced? What enzyme are we talking about? How many times is it used per period of time, per unit time? All those things have to be factored in. Those are questions that you would have to have more information than what we have at this point in order to know that answer. Okay, so they speed up the reaction, and they lower the amount of energy required to make that reaction. They lower the amount of energy that is necessary to make that reaction occur. So that is what you call energy of activation, the amount of energy that you have to put into a reaction to make it work. And I said before, well, okay, if I'm in Miss Jackson's chemistry class and I put molecule A and molecule B into a test tube, it'll probably just sit there. Nothing's going to happen. But I have to put it over a Bunsen burner. X amount of heat or X amount of energy. It doesn't have to be heat. It could be electrical energy. It could be any number of different energy sources in order to get that reaction to occur. We tend to think of heat, as energy. Well, it's just one type of energy. But, if you have an enzyme there, you don't have to add as much heat. That's why living things require those enzymes, because we can't possibly work. Our cells couldn't possibly function at the temperature of a Bunsen burner. It isn't going to happen. In order for our body temperature to get that hot, in order to speed up all the reactions to make them work, we'd just melt. We couldn't possibly do it. So we have to have something in there that will lower that amount of energy to a point where it will work, but it will work at 37 degrees Celsius, 98.6 degrees Fahrenheit. It has to work at that temperature. Sometimes biochemistry, chemistry of the cell, and biology, life, is sometimes called warm chemistry, which is what it is. It's like walking into a chemistry lab, a PChem, physical chemistry. It's just warm chemistry, and that's pretty interesting. I like that. So this next slide, what you're seeing here is the amount of energy. I shouldn't click that twice. I'm an idiot. I know better than that. I have to click on this one, they're showing without the enzyme and with the enzyme. Okay. So if I don't put an enzyme in it, then I have to have this much energy. I have to go way up here. So the black line shows me, I don't want to use that. I want to use that. So this black line right here shows me I have to put in that much energy in order to get that reaction to occur. All right. But if I put in an enzyme, I don't have to put in that much heat or electrical energy or whatever. I only have to put in that much energy. All right. So I don't have to add as much energy. I lowered the amount of energy required for the process. So yeah, that's what they call, that isn't going to work. That's what they call energy of activation. How much energy is required to make that reaction occur? And we don't add as much as long as you have enzymes. So biological systems, yeah, they go ahead and they have to have enzymes. There's no choice. We can't operate at Bunsen burner temperature. I don't think that'll work. Okay, and then of course whatever we're reacting, if you have an enzyme, whether it's two molecules adding together to make one, or you have one breaking down to make two, those things are the substrates. We have substrates. And of course, if we don't have an enzyme, then they're called reactants. If you do have an enzyme, they're called substrates. Big difference. Because in chemistry, you just work with reactants. In biology, you work with substrates because we always deal with enzymes most of the time. Most reactions need enzymes. I'm not saying all of them, but a good many of them do. I'd say at least 90%, and I'm sure someone has a figure out there somewhere where they've gone, well, it's got to be at least 98%. I don't know what the figure is. Google it, see what you get. Okay, so the active site is where the molecule actually enters, and we've been through that. And they're very specific. Yeah. So if I have a particular cell, it's only going to be able to produce certain enzymes because the function of that cell is very specific. The pancreas cell has the ability to produce enzymes for digestion. All right. The cells of the intestine have enzymes. The small intestine, at least, it's actually broken into three units: the duodenum, the ileum, and the jejunum. You got no. Duodenum, jejunum, and then ileum. Ta-da. Okay, so you got three pieces there, but it's all one long tube, about 21 feet in length together, all together. But when you get into past the duodenum, which is really only 10 inches in length, that's where you do most of your digestion. People go, we digest in the stomach, and tell everybody they're wrong when people tell you that. We eat food and we digest in the stomach. Just say, no, wrong. We do a little bit, very, very little digestion there. Not much at all. So most of it happens in the first part of the small intestine called the duodenum, duodenum, who cares, but duodenum. And that's just 10 inches long. And so for a male, that would be about like that. For a female, it would be 10 inches. It's a joke. Maybe not a real funny joke. Okay, but I think it's funny. I love to laugh. Anyway, so yeah, they're very specific. So if you're in the duodenum, you know, you've got all these enzymes going on there to do all the digestion. But once the material is digested and moved further down into the jejunum and the ileum, you have enzymes for transporting the things that you digested into the blood. So you make a completely different set of enzymes because you want to transport this amino acid, this amino acid, this amino, all 20 of them. You want to have enzymes to transport all of them into the blood. You know, you want to transport this sugar, this sugar, this sugar. You want to have enzymes to get all those sugars. You want to have enzymes to get your fatty materials. You know, you want to have enzymes to get your vitamins that you've taken, your minerals, blah, blah, blah. You know, so you're very specific depending on what the cell is. And I'm just talking about the small intestine. You know, I mean, it's really all we're talking about there. So, you know, you can imagine you go through the entire body and how many different enzymes are going to operate the eye and how many are going to operate, you know, the lining to the nose and how many are going to operate depending on where you are. It's very specific is how that works. So we've done this, but here's the enzyme. It has an active site. Now, notice on that active site, it is an exact fit. That's why they call it an induced model. So that enzyme really kind of looks like, I don't know, it certainly doesn't look like this substrate, sucrose, which is a disaccharide, 2 sugars. One is a hexagon and one is a pentagon, 5 sides. There are 2 pieces to it. One is glucose and one is fructose. But they certainly don't look like this active site of this number 1, the enzyme available with the empty active site. But once that substrate, sucrose, goes in there, then you get an induced fit model. It fits it exactly. The enzyme changes shapes. It goes in there. You got a perfect fit. All right. So, and in this case, we're going to break sucrose into 2 units. You're going to break it into the glucose molecule and the the glucose molecule and the fructose molecule. You actually break it down. So that's a degradative reaction. So we end up getting both products, the glucose and the fructose, coming out of that. And then, of course, you release the enzyme. It can be reused to a point. Obviously, it will wear out, and you won't be able to use it forever. You have to make more of them. Okay, so there are optimal conditions under which that enzyme works. We dealt with that yesterday in lab. They work at specific pHs. You know, and so you go, well, enzymes must work best at a neutral pH. That would stand to reason. Neutral pH. Well, the enzymes of the stomach work at a pH between 1 and 2. That is really, really, really acidic. Very acidic. If you've ever vomited, that's a lot of acid. You're just like, I do not want to vomit because that is really nasty. And what's so nasty about it? You know, don't look at it. I don't look. I'm not going, oh, look. Look at what I found in my vomit. Look at that. Yeah, I remember eating that thing. Yeah. How many weeks has that been in there? That's a joke. At a very oh no. Hello. Hello. Testing. We do have sound. Okay, continue. Hello. Hello. You should have sound. All right. So everything operates at specific pHs. Stomach, pH 1 to 2. Duodenum, pH 8. Blood, pH from pH 7 to pH 7.8. It tries to stay at about 7.4. Depends on where you are. Very specific pHs for very specific enzymes depending on where they are. Temperature, most of our enzymes will work at our body temperature. If you're a rattlesnake, you have to crawl out of your hole in the morning. You get out on the pavement because it's black and it heats up really fast, and you're out there just soaking up the heat until you can get enough heat to make enough energy in order to move, to find food. That's really what you need to be able to do, or a mate. It's one or the other. That's really the whole life of a the purpose of that animal, you know, they either need to find food or find a mate. What? That's about it. You know, snakes have a hemipenis. They actually have two. So that when it goes into the female and becomes erect, it goes like that. So she can't get away until he's finished, you know. It's like, get off me. And he's going, you can't move. I'm gonna be done here in a minute, you know. Anyway, sounds pretty weird, but it is the way it is. That's the host snail thing video that y'all have got to watch. I will send that to all of you because it's too funny. They're hermaphroditic, so they stick their penis in each other's vaginas, you know. And the guy who's talking about this stuff is just so funny. He is so funny. And it's different things. They have darts, love darts. They shoot at each other. You know, I mean, seriously, love darts. And he goes, man, they take kinky to a whole new level. Like, really? You didn't just say that. Yeah, he did. Okay. These are some things that some enzymes need and some enzymes do not need. These are plus or minus situations, basically, what I call plus or minus. Plus or minus. On the plus side, that would be cofactors. These are what we call minerals. So again, some enzymes need them, some do not. These are minerals. That would be such as sodium. You've heard of these: magnesium, iron, potassium, and they're not all positive. Chlorine, blah blah blah, a bunch of them. Coenzymes. These are what we call vitamins. So, A, B, B1, B2, B3, B6, B12, blah, blah, blah. Ta-da. Anyway, there are a whole bunch of them. And C's and D's, you know, and E. You know, and once they started finding all these vitamins, they went, wait a minute, this isn't going to work. We only have 26 letters. We keep finding more and more and more. So instead of using the ABCDE system, the alphabet, they started naming them by the chemical names, like ascorbic acid, that would be vitamin C, and thymine would be vitamin B12. No, it isn't. I think it is. I could be wrong. Doesn't matter. But anyway, so they started giving them chemical names all of a sudden. But so some vitamins require cofactors, minerals. Some require coenzymes. Some don't require either. And some enzymes require both. So you really got to know your enzyme. Need a cofactor and a coenzyme, or does it not need either, or does it just need one or the other? And which one does it need, the cofactor or the coenzyme? Depends on the enzyme as to what they'll need. So those are pretty good examples of those. Okay, I'm getting really close to the end, but that doesn't mean I won't stop right before the end. Because this lab is really going to require some orchestration, I've set this thing together. And let me explain, because I'm so excited to get to do it seriously. Because if you listen to the pre-lab, and I hope you did, if you listen to the pre-lab, you'll go, really? You're going to try to do all this in an hour and a half? Yeah, because what I'm going to do is we're doing 2 labs in 1 hour and a half period. Normally, that's 2 weeks of lab, 2 whole weeks. Okay, so how in the world are we going to do this? Well, you are going to only be responsible for doing this one thing with this one test tube. You're going to only be responsible for doing this one thing with this one test tube. And I've divided it up, and I've even put the equipment that you need for your specific activity on your lab table. So, you know, it's like, okay, you guys are doing this, you guys are doing this, you guys are doing this, you know, and I broke it all up and set it all up, and all you have to do is one specific thing. And it can be done literally in a period of 45 minutes, all of it. And then once it's all done, then we can just throw all our test tubes away, drain whatever we have in them, pour the test tubes away. Except for one thing, we don't want to put that down the drain, and I'll tell you which one that is. But and then we'll talk about it, what was supposed to happen. And I actually made up a lab review sheet because I don't think we're going to have time to go through a review. So I made up a lab review. Now, I think if you go home and you go, first question, all right, here's the answer to that. Second question, here's the answer to that. When you get to the test, first question, guess what? It's the answer to the first question that you had on the answer sheet. Second question, guess what? It's going to be the answer to the second question you had on your review sheet. Third question on test is the third answer to the question on the review sheet. And so, bam, 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 you basically have the test, is what I'm telling you. You just have to make sure you've got all those answers. And so I've been really, really kind with it, but at this point in the semester, you guys are you're getting beat down. I mean, I don't know it. I see it. I look around the room, and believe it or not, I can tell. You are beginning to go, Mother Bear. This is really starting to tax me. This is real. I'm getting really tired of having to work this hard for this period of time for 8 weeks. And I understand that because we're all having to work that hard. Absolutely. Not just you guys. So you know, I'm going to give you a break. I'm going to give you a little bit of a break. And that's cool. But I've orchestrated this thing, and I worked it out in my mind last night, and I was just like, okay, cool. If I can pull this off, I will be like doing backflips. I've already shown some people in the department. I said, come in here. I want to show you what I'm doing. You know, so this table's good, and this da-da-da-da-da-da-da. And they go, take a photo of that. So I took a photo of the whole lab, you know, and I'm going to see if it all works like it's supposed to. And then we get together when everybody's finished, and we go, okay, let's talk about this and this, and, you know, what did you get? You know, what happened with yours? What happened with yours? And blah, that kind of thing. And hopefully it will work. I've got faith. You know, if any class could possibly do it, it's in all